in third year, because this is a course that will be taught in both second and third year. So this is the outline. I'll just give a brief history. I know you discussed a few things. A few aspects of the immune system in your last physiology lectures, but we'll go into more details. We'll discuss the concepts of the immune response and what happens when a person cannot develop a healthy immune response and we end up with immune pathology. So immune pathology will be the brunt of most of what we will cover next year. So the function of the immune system is basically to protect the host, the multicellular organisms like the human being, from pathogens. And you'll see from your microbiology lectures that there's a diverse array of pathogens ranging from very small viruses, which cannot be seen with the naked eye, to large helminths or parasitic worms. And for us to be able to provide defense against this wide array of pathogens, our immune system needs to be able to recognize them and have different mechanisms of destroying them. So the immune system consists of a dynamic network of cells, molecules, like soluble molecules, and molecules expressed on the surfaces of these cells, and pathways that interact with each other so that you can have that immune defense. So a bit on the historical perspectives of immunology and how the concept of immunity came about. So a long time ago, some smart people were able to realize that individuals who had recovered from certain types of infectious disease were later protected from that disease. For example, if there was an outbreak and they had recovered from that particular disease in a previous outbreak, then in a second outbreak, they never developed that disease. And that is how they coined the term immunist, meaning exempt, and that's where the term immunity comes from, which basically describes a state of protection from infectious disease. More information was discovered from early vaccination studies. Vaccination is the deliberate induction of immunity. As you well know, I'm hoping you've all received your COVID vaccine, so that even if you encountered someone who's infectious, who's harboring the pathogen and is infectious, you wouldn't develop, either you wouldn't develop disease or you wouldn't develop severe disease. So that's the element of deliberate induction of immunity. So early vaccination studies began in the 15th century among the Chinese and the Turks with an attempt to prevent smallpox. The picture there shows a boy suffering from smallpox, and you can see those really large lesions that heal with scarring. So what they did, they would get crusts from these skin lesions and either inhale them or insert them into cuts in the skin. And the hope was that someone would now maybe develop mild infection, but they would evoke an immune response, which would prevent them from developing a severe infection on future encounter with a pathogen. So that was called variolation. This continued later into the this is the 16th or 17th or 18th century, I forget. Well, in 1718, there was this lady, British lady called Lady Mary Montagu. She had observed what the Turks were doing with the variolation, and she had it performed on her children. So in that era, smallpox would really cause ravaging disease, especially in forms of outbreaks, and she wanted her children protected. So she had this performed on them. It's 18th century. So in the late 18th century, I'm sure you've all heard about this physician called Edward Jenner, somewhere for those of you who read widely. He observed that milkmaids who had contracted cowpox were subsequently immune to smallpox, to severe smallpox. So he figured or hypothesized that if you inoculate fluid from the cowpox pustule, so this is from the cows, into people, so similar to the variolation, but now you're taking pustules from the cow instead of from an infected human, and you inoculate that fluid into humans, then they would be protected from severe disease. So he tried this on an 8-year-old boy and then later infected him with smallpox, and he was able to confirm his hypothesis. And then this technique later spread out through Europe to vaccinate people. And then came this other French scientist, Louis Pasteur, who did a lot of work with bacteriology. And he was able to isolate or grow the bacteria that causes cholera amongst chicken in culture. And in microbiology, you go through, I think one of your lecturers will mention the Koch postulates and, the several, the criteria that are required for one to, say that this particular pathogen causes this infectious disease. So for example, you should be able to isolate it from someone who has that infectious disease by growing it in culture. And if infected in, for example, even if it's a lab animal, then it should produce that particular disease. Those are some of the criteria. So he was able to isolate this bacteria, and then he confirmed that when he injected it into chicken, they would develop cholera. Okay. So he later also re-injected chicken with an old bacterial culture, so a culture that had been in the lab for a long time. And he realized, so with the fresh culture, the chicken developed cholera that would actually cause mortality, so it was fatal. But when he did the same thing with the old culture, the chicken did become sick, but they recovered. And then he later grew a fresh culture and injected, so he was doing an experiment, a mix of chicken that had previously been injected, the ones that were injected with the old culture and recovered, and an exposed chicken. And he realized that with the fresh culture, only the fresh chickens died. So what he was able to establish is that the aging of the culture, the bacteria in the lab weakened their virulence, their ability to cause severe disease. And this is where the aspects of administering weakened, or what we call attenuated vaccines, live vaccines, but they're weakened, their pathogenicity is reduced. This is where that practice came from, to provide immunity against a disease. And when we cover the vaccinology series, you'll later come to see that the live attenuated vaccines provide better immune protection compared to killed vaccines or just subunit vaccines where you're only taking a part of that pathogen. So he called, this is where now the term vaccine came from. He coined this attenuated strain a vaccine, and this came from the Latin word vacca, which is a cow. He later went on to work with Bacillus anthracis. This is the bacteria that causes anthrax in animals, and he vaccinated a group of sheep with Bacillus anthracis that he had attenuated by heating, and he vaccinated both. He challenged the sheep he had vaccinated and another group that he had not vaccinated with a virulent culture. And of course, it's the vaccinated sheep that did not develop disease, while the unvaccinated ones died. Just going further to confirm how vaccines work and how they provide protective immunity from severe disease. He also did some work with the rabies virus, where he administered a series of weakened virus preparations to a boy who had been repeatedly bitten by a rabid dog. And the boy was able to recover and not get severe rabies, which causes really severe disease that ultimately would be fatal if nothing was done. And this boy later went to work in his Pasteur Institute, which still exists up to this date. So the purpose of this was just to show you that the study of immunology was linked to the discovery of vaccines. Like the two went hand in hand, defining how do we get protection from infectious disease. Okay. It is a fact that vaccination has aided in improving mortality rates worldwide, especially amongst young children under 5 years of age, because they really don't have very strong immunity. And I know you all are recipients of what we call the childhood vaccines. And all these vaccines are just to protect against infectious disease, which, if they were not available, would cause a lot of mortality in children of that age. So, your measles vaccines, mumps, rubella, polio, tetanus, and so forth. We do not have vaccines against all infectious pathogens. I know there's a malaria vaccine that was developed recently, but it doesn't, in terms of providing protective immunity, it's not very effective. It does provide some protection, but it's not very effective. So there's still work going into that. A lot of work going into developing an AIDS vaccine as well. And maybe Professor Anzala in microbiology will tell you more about that and the work they've been doing at the CAVI, Institute of Clinical Research. CAVI stands for Kenya AIDS Vaccine Initiative, although they've expanded into more research, but initially started as an institute to just try and get an AIDS vaccine. They've performed quite a number of clinical trials, but we haven't been able to successfully find an AIDS vaccine that will prevent infection. When it comes to vaccination, there's some diseases, there's possibly only one that has been eradicated by vaccination, and that is smallpox. The last known case of naturally acquired smallpox was documented in 1977, and this was a patient from Somalia. So since then, there's been no documentation of a smallpox case. They're trying to do the same with polio to try and eradicate it. And that is why you see campaigns. I think there was one actually recently this year. Every time there's a wild type polio, meaning natural infection detected anywhere in the country. Usually this happens around the borders, and especially from Somalia. Then the government does a campaign to revaccinate all children under 60 months or 5 years. There's the concept of herd immunity, which I'm sure you've had people talking about with this SARS-CoV-2 pandemic, which basically refers to the fact that you want a critical mass of people to have protective immunity against a certain infectious pathogen. And this protective immunity can either come from natural infection or from vaccination, so that you're able to decrease transmission to people who are susceptible. Maybe they haven't been vaccinated, so they're susceptible, and there's a reason. Maybe the vaccine is contraindicated in those people. And so you're basically preventing transmission of the pathogen, okay? So immunology is more than just, the science of immunology came into play when people were, trying to really describe or define protection from infectious disease. But immunology is about more than just vaccines and infectious disease. In the current era, there are people who have been able to manipulate the immune response in several ways, either to boost a response. For example, in cancer immunotherapy, you really want to you're treating the patient with, for example, chemotherapy or radiotherapy, but you also want to utilize the immune system to try and fight those cancerous cells. So that's immunotherapy. The immune response can be manipulated to inhibit it. For example, in patients who have diseases where it's the immune system that is responsible for the pathology, like in autoimmune disease. Or you can even modulate the efforts of the immune system just so that you can get some relief. So a lot of application in autoimmune disease, in allergy, in cancer, in transplantation, and so forth. So what is it in the immune system that is working, and how does it work exactly? What are the mechanisms of immunity? As much as Louis Pasteur was able to show that vaccination worked, he couldn't really define what is it that is working, okay? So there's a German scientist who was later able to demonstrate that it is cells. There are some cells in the body that are actually responsible for immunity, and he was able to observe them under the microscope, rudimentary microscopy back then, and he was able to see these cells ingesting microorganisms and other foreign material, which is what we call phagocytosis. Okay, and then in the 1890s, this German scientist von Behring and Kitasato from Japan, and I think they are Nobel Prize winners, demonstrated that it is actually something soluble in serum. So it's the serum which is responsible for immunity. They were able to demonstrate that if you take serum from animals that had been immunized with diphtheria and transfer that serum to unimmunized animals, then those unimmunized animals now became protected from diphtheria. So that's when the two concepts came into play. One group thought it's cells that mediate immunity. The other one thought that it's sera. And this is where the concept of cellular mediated and humoral immunity came into play. And we'll discuss this further. So for humoral immunity, we're talking about, soluble mediators in the blood. Okay. And what really drove the discovery of this is, the realization that there was something in this sera that could either neutralize or precipitate toxins. So prevent these toxins from pathogens from attaching to their cognate receptor in the host, so that the toxin can become active. I'm talking about things like the tetanus toxin or the diphtheria toxin. There are certain pathogens whose pathogenicity is because of the toxin they produce. The toxin they produce is the one that causes disease, the cholera toxin, for example. And they also realized that this soluble mediator in sera could cause bacteria to clamp, what we call agglutination. So they named these components according to the function, the effector function that was seeing. Okay, if it was able to neutralize or precipitate the toxin, they called it an antitoxin or a precipitin. If it was able to agglutinate bacteria, they called it an agglutinin. In the early 1930s, there's this scientist who was able to fractionate serum, or discover what the different components of sera were based on electrophoresis. And he discovered that there's a component called the gamma globulin, based on how they were able to separate the different components according to size using an electric charge. And you'll cover a lot of this in clinical chemistry. So the different fractions that were seen when the protein separated were named alpha, beta, and gamma. So he identified that this fraction called the gamma fraction is the one that was responsible for this antitoxin or agglutination activities. So they later called them antibodies. Okay, so these names are interchangeable: antibodies or immunoglobulins or gamma globulins. Okay, and why this is called humoral immunity is because back then body fluids were called humors. So depending on where it was found, it was still called a humor, and we've maintained that terminology of humoral immunity. So after this discovery, people were actually able to perform some rudimentary immunotherapy, where they would take the serum from animals. Usually they would use horses because they have a large amount of blood, and want to use the antibodies in that sera that's been pathogen exposed to administer to patients suffering from certain diseases. So for example, they'd vaccinate that animal with either diphtheria or tetanus, and then they harvest the blood and isolate the serum. Okay. There's current therapy where we take advantage of immunoglobulins to protect individuals against certain diseases. For example, the snake antivenom. Is just based on, using pooled immunoglobulins to treat snakebite victims. Later, the terms passive and active immunity were coined. So passive immunity refers to immunity where you're administering immune components to a host, already formed immune components. So the host is not forming their own. For example, the anti-serum. So these are normal, biological products. They're proteins. They have a half-life. Okay, they're not going to live forever. So this type of immunity would be short-lived. Okay, as opposed to active immunity, where you're actually administering an active component of a pathogen. Okay, probably it's been weakened, so that the individual can produce their own immunity. They produce their own specific cells and their own antibodies. And this can be active immunity is obtained either through vaccination or from natural infection. I don't know what Daniel means. Is this a question? When your body's antibodies mistake normal proteins in the body as foreign antigens and start attacking them? I'll allude to this. Okay. Okay, and then the concept of cell-mediated immunity, now that they discovered there's some white blood cells that are able to ingest pathogens. There are many people who worked on this and finally came to discover that there's a certain cell called the lymphocyte, which is the one that is responsible both for cellular and humoral immunity, meaning that these cells, these lymphocytes, are cells that can actually attack pathogens, as well as being the cells that are responsible for producing these soluble mediators that we are calling the antibodies, okay? And later on, they were able to discover that there are two types of lymphocytes based on experiments in chicken. And these were defined based on where they were derived from or developed from. So the T lymphocytes and the B lymphocytes, which were isolated from the thymine of this chicken and from the bursa of Fabricius. And the same applies to humans, except that the B lymphocytes come from the bone marrow. So you can still remember the B for bone marrow and T for thymus in terms of where they mature. So in summary, the take home here is that cellular immunity is imparted by T cells, and we'll see how the mechanisms of cellular mediated immune responses. And then humoral immunity is imparted by B cells through the production of antibodies, okay? And they really do not work independent of each other. Both are needed for a complete response against most pathogens. And we'll come to see how important that is and how they interact, how you cannot separate cellular immunity from humoral immunity. The other aspect we will discuss further is what does the immune system recognize, okay? And there are several terminologies that will define further, for example, your antigens, immunogens, and haptens. But basically what they recognize is what we call an antigen. And an antigen is a substance that elicits a specific response by either B lymphocytes or T lymphocytes, okay? And it can interact with those cells. For example, it can bind to a receptor on the T lymphocytes. It can bind to either the B cell receptor or the antibody that is produced by the B cell, okay? I think I will skip the rest of that slide and just finally just show you the basic outline of the immune response to the cellular and humoral mediated immune system and how both are induced into effector function once there's exposure to an antigen, which could either be an infectious agent or any other foreign protein. So both will elicit a humoral and a B cell response. So this antigen can bind to a B cell receptor. You get the B cell being activated. It differentiates into a cell we call the plasma cell. So the plasma cell is the one that actually secretes the soluble antibody. And this antibody has the same specificity for the antigen that initially bound to the B cell receptor. So the B cell receptor is also an antibody molecule. It's only that it's expressed, it's surface or membrane bound, but it's the same molecule as what will be secreted as your immunoglobulin or your antibody. And then it will get rid of the pathogen by several mechanisms. That same antigen will also activate your T cell response. However, antigens are not able, like T cells are not able to recognize antigens directly. It has to be presented to it by what we call the antigen presenting cells. That is not really shown here, but we'll discuss this in further detail. And once the antigen presenting cell presents this antigen to the T cell, it becomes activated. There are two types of T cell. There's one we call the T helper cell, which usually secretes cytokines. Cytokines are soluble mediators, and they're like messenger molecules that communicate with cells, and they exert their function by binding to a receptor on these cells. So these cytokines can also activate your B cells to produce antibody, and they can activate your other type of T cell, the cytotoxic cell, to become more effective in killing, activate it to perform its function, which is to kill infected cells. Okay. Okay. In terms of protection against immunity, against infectious pathogens, I've already alluded to the wide array of infectious pathogens, and the process by which they induce illness is what we call pathogenesis. You'll discuss a lot of pathogenesis and pathology in your general pathology lectures. The major groups of pathogens are your viruses, fungi, parasites, and bacteria. Okay. They all differ. For example, your viruses are really small. This is an electron microscopy picture, and you can see if you compare the size of your virus, for example, to the size of a helminth, they're really quite small. Okay. So this is a rotavirus as an example. You have your fungi. For example, the Candida albicans. This is a host cell, and this with the spore and the hyphae is your fungal pathogen. This is a filarial worm. They're much smaller than the other helminths that we see that cause, for example, like your ascaris or your roundworms. The filarial worms are quite small, and this usually are found in the blood. And then this is a rod-shaped bacteria, a bacillus. Okay, this is Mycobacterium tuberculosis, and this is just to show you the size relative to a host cell. This is probably a phagocyte that is trying to ingest a clump of bacilli. So they're even much smaller than our immune cells, except for the helminths, which are larger than the host cells. The rest are really small pathogens. You need a microscope to be able to see them. For the viruses, not just a light microscope, but an electron microscope. Okay, so our immune system needs to be able to recognize the presence of these pathogens in our body so that it can mount a response and get rid of them by destroying them. Okay. And the type of immune response that will be elicited also depends on the microenvironment of where it is. For example, areas like the central nervous system, like the brain, you don't get a lot of immune responses just because that immune response itself may end up causing more damage than the presence of the pathogen. Okay. And there are certain aspects of what we call tolerance where the immune system may not really mount a response depending on where the pathogen is. For example, we'll come to describe what we call the normal flora or the microbiota or the different microorganisms we have in certain parts of our body that are actually not harmful if they're in that region. For example, some of the bacteria that we have within the gastrointestinal tract, we tolerate them when they're there. But if they invade another part of the body, for example, E. coli within the genitourinary tract would cause infectious disease, or if it gets into the bloodstream, it causes sepsis. But within the gastrointestinal tract, depending on what exact isotype, then usually we'll tolerate it. And the other thing to remember is that for a pathogen now, or for the immune system to be activated, recognize a pathogen, it has to invade the physical barriers of the body. And that's why the physical barriers are, part of what we call the innate immune response. They're already there and they provide protection. So things like the skin and the mucous membrane, okay? As long as your skin is intact, then a pathogen cannot come in through the skin, for example. And then we have other barriers like some enzymes in the saliva, actual mucus within these mucous membranes, like within the respiratory tract, acid in the stomach, and the vagina. The vagina, I would rather say the low pH in the vagina, and so forth. Also provide protection against invasion by these pathogens. So in terms of recognition of these pathogens, this involves interaction between certain molecules on the surface of the pathogen and certain molecules that are expressed by the host cell, okay? So the ones on the host cell, we're going to call them receptors, okay? These receptors can either be bound on the host cell or they can be secreted. So they could be membrane-bound receptors or soluble receptors that can interact with specific parts of the foreign organism, okay? And initiate recognition, and then you get an immune response. Okay, so what is on the pathogen or what is being recognized is what we call the ligand. Okay, so you get interaction of the receptor and the ligand. So these ligands for recognition could include the whole pathogen, some specific fragments of the pathogen, okay, or products that are secreted by the pathogen. For example, a toxin would be a ligand, but it's something that is secreted by the pathogen. An antigenic fragment would be something like the pili of some of these, which are like hair-like structures of some of these pathogens, or flagella that help them move, okay? So the outcome of interaction between the ligand and the pathogen would be now a cascade of events that leads to labeling of that pathogen. So it becomes a target, so it becomes labeled, and the immune system knows, okay, we need to get rid of this thing, and then it becomes destroyed. So that is technically what the immune response is all about, okay? So I don't think I'll go into details of this. Was an example of the destruction of viruses, which usually cause intracellular infection. I'll skip that for now and just describe these ligands further. So what is recognized on this pathogen is what we call, what we called pathogen-associated molecular patterns. So these are chemical structures that characterize these pathogens. And, they're just like arrangements of, for example, the carbohydrates and other molecules on the surfaces of these pathogens that are very different from what we have on our host cells, okay? For example, if you have some of the capsulated pathogens, okay, or capsulated bacteria like Streptococcus pneumonia, that capsule and the arrangement of the different molecules, like the amino acids, for example, not amino acids, it's a carbohydrate. The polysaccharides on that capsule is very different from what we have on our host cells. So that is how and receptors. And it's usually our white blood cells that recognize these particular patterns, okay? And once they do that, then these pathogens are marked for destruction. So these pattern recognition receptors that we have on our white blood cells, they're the same on all white blood cells, okay? And they're encoded in the germline, they do not change, okay? So they're always the same, okay? Why I'm highlighting that is because you'll come to see the variability in the T cell receptor and in the B cell receptor, okay? Those ones are not like two clones of T cell receptors or two clones of T cells do not have the same T cell receptor in terms of specificity of what they will interact with, and the same for the B cell receptor. Okay, you'll discuss that further in your lectures on the ontogenesis of the B and the T lymphocytes. And that's is what brings about diversity of the immune response. So B and T cells are able to recognize virtually any antigen possible just because of the variability in the antigen binding portion of their receptors, which is different from what we have in our other white blood cells. Okay, the non-lymphocyte white blood cells. Okay, and this diversity comes from rearrangement of the genes that code for these B and T cell receptors. Okay, you'll discuss that further in your ontogenesis lectures, as I've said, so I'm going to skip that and bring in another aspect that is important to know and will be described further, of what we call tolerance. Okay, so tolerance, I mean, it just means the same as the English word. When you tolerate something, like something is there, but you're not getting provoked by it. So this is an aspect of immune non-responsiveness to certain types of antigens. And the importance of tolerance is just to ensure that the immune system does not accidentally recognize and destroy host tissues. You know, you don't want your immune system to mount a response against your self antigens, okay? And it relies on what we call self versus non-self discrimination. And it's the B and T lymphocytes that take part in tolerance, okay? So in ontogenesis, and we'll mention it when you have a specific lecture so that they don't, in future, after they mature, mount a response against self-antigen. They'll know that says this is a foreign cell or a foreign antigen, it needs to be destroyed, okay? The downside of tolerance, of self-tolerance, is that now, you know, when you have a cancerous cell, really that is a self cell, but it's a harmful cell. It's not dying, okay? Then the downside of tolerance is that the immune system usually will not mount a very strong response against cancerous cells, even though these cancerous cells do express certain antigens that we call the cancer-associated antigens, which we'll discuss next year, okay? When you do not have tolerance, or there's a problem with tolerance, then that is when we get autoimmune disease, you know, disease against self. You get immune responses against self-antigens. The other important aspect that we'll describe further is the innate immunity. As I've already alluded to, this is, you know, what is inbuilt and already encoded in the germline. It's already present. So it's usually the first responder. It's very primitive, you know, it doesn't become specialized with time. It doesn't become more effective with time. But the importance of the innate immune response is to prevent infection immediately or quickly. Okay, and this includes things like I've talked about physical barriers, chemical barriers like low pH, your cells with their pattern recognition receptors. So this usually would be the phagocytes. Okay, and then we do have some soluble components in serum that are aspects of the innate immune response. For example, there's a series of serum proteins called complement. Which usually what they do is like when complement proteins are activated, they initiate a cascade. You get a cascade of new proteins being formed. And the ultimate end effect is that you get lysis of either infected cells or of pathogens, okay? But you also get other events being activated like inflammation and so forth. We'll have a whole lecture on complement. And then the other component of the immune response is adaptive immunity, which, as the name suggests, it adapts with time, it gets better with time, it gets more specialized. And adaptive immunity is mediated by the B and the T lymphocytes, okay? It takes time to be activated, so it's not the fast responder like the innate immunity. So it takes about, 5 to 7 days, but it is very specific. Okay. So your phagocytes will ingest or recognize and ingest any pathogen. Okay. You won't have like a specific phagocyte that will only recognize this type of bacteria, for example. But for your B and T cells, once they're activated, they can only the immune response can only be targeted towards the antigen that activated it in the first place. So it's very specific. It evolves with time, so it becomes better with time. Okay. As I mentioned, it's a second line of defense. So when it comes to vaccination, the goal of vaccination against infectious disease is to elicit development of specific and long-lived adaptive responses. Okay. The other aspect of adaptive immunity is that it has memory, what we call immunological memory. This describes the ability of the immune system to respond even better and quicker during a second exposure to the same pathogen. Okay. So, and here we're just discussing the adaptive immune response. The innate immune response does not have memory. Okay. So when we're discussing immunological memory, it's good to know that on first exposure to a certain pathogen, when you get your adaptive response activated, we call this a primary immune response. So you activate, a few clones of the lymphocytes that will become specific against that particular antigen and get rid of it. On a subsequent exposure, and this is also another aspect we take advantage of, or we try to manipulate with vaccination, first dose versus second dose of a vaccine. So on subsequent exposure with the same antigen, what we get is a secondary immune response. So here, you're not starting the whole process of activating, new lymphocytes. The same lymphocytes that were activated will produce what we call long-lived B and T lymphocytes. So these are the ones that have now become reactivated to fight again. Okay, so the process of, or rather initiation of the secondary immune response doesn't take as long, the 5 to 7 days, as the primary immune response. This table just compares the innate and adaptive immune response in terms of time. Innate is immediate, so minutes to hours. Adaptive takes a few days. In terms of specificity, the innate one is limited and fixed. The adaptive one is highly diverse and keeps improving. Okay. In terms of response to repeat infection, innate immune response is always the same, but the adaptive immune response, that's why you have immunological memory. So a second response will be quicker, it will be more effective, and a third one will be even better. It just keeps getting better with time. The major components for innate immune response include things like your physical barriers, chemical barriers, and in terms of cells, it's your phagocytes with their pattern recognition receptors. For the adaptive immune response, the components are the T and B lymphocytes with their antigen-specific receptors. So that's the T cell receptor and the B cell receptor, and the antibodies that are secreted by the B cells. This graph just shows us, the difference in terms of time between the adaptive and innate immune response. So you get exposure to an antigen. On the x-axis, we have the time in terms of days, and on the y-axis, this is a measure of the magnitude of the response. So the innate immune response becomes activated immediately, okay, and peaks. But the adaptive immune response, you see there's a lag in the activation, so about a week before it picks up, okay? And then within a month, both go back to baseline. That's the primary response. So if the second exposure to the same antigen, the innate immune response will just be the same, it's activated immediately, it peaks and tries to get rid of the pathogen. But now you can see on second exposure, the adaptive, because of memory cells, adaptive immune response peaks almost immediately, okay? And then you can see the steepness of this curve. You get a better, so it's a quick response, and it's even of more magnitude than the fast response. And usually this will not go back to baseline. For example, if you have specific antibodies produced, they will persist for some time. Finally, the pathology of the immune system. What happens when you cannot develop a healthy immune response? okay, which I will just introduce because we may allude to some of these things, but most of this we will discuss next year. So you can get inappropriate or dysfunctional immune responses. In physiology, what you covered was, the normal body processes and homeostasis. So even for the immune system, you need a balance. Yes, you need a very good response against pathogens, but when the pathogen has been gotten rid of, for example, you need to stop that response. So there always needs to be regulation. And we also need to ensure that the immune system is not attacking something that it should not. So it shouldn't attack cells. And it should not fail to attack something it should. You don't want it not to be able to mount a response against pathogens. And there are several categories of diseases that describe these dysfunctional responses, and we'll discuss them. One is your hypersensitivity responses, where you're getting very exaggerated attacks on antigens that are foreign, but they're not really infectious, okay? And this is basically defined by, a good example of this would be an allergic reaction, where, someone is mounting, if it's a food allergy, a very strong immune response against food, which we should be able to tolerate, what we call oral tolerance, or against pollen or dust mites, okay? So it's this response that now causes symptoms or causes disease. The other aspect is autoimmune disease, where you get the immune system erroneously mounting a response against self-proteins or self-tissues. And then immune deficiencies, where the you don't have an immune response against infectious agents. So usually the patient will manifest with recurrent infections because they're just not able to get rid of them, okay? The other aspect of, I would say, the pros and cons of the immune system is in transplantation. So transplantation is the science where you want to replace a non-functional organ with one that is functioning. And usually you get this organ from a donor, for example, if it's a kidney. So if someone has end-stage renal disease and their kidneys are not working, depending on the age, there's a balance between, okay, they're going to come for dialysis 3 times a week for the rest of their life, versus you can actually transplant, give them a kidney, someone else's kidney, because we have 2 and you can actually live well with 1. But the problem is the immune system will be able to recognize antigens on that kidney that are not self, they belong to another human being, and can mount a response against that, and you get rejection of that organ. So what usually happens is that you would have to place this patient, the organ recipient, on, suppress the immune system with drugs so that they're able to tolerate that kidney. But you have to achieve a balance where you're suppressing the immune system so that they don't reject the kidney, but you don't want to suppress them too much that now they're getting infections. Okay, so that's one of the major challenges seen in transplantation. I've alluded to malignancy, and you'll discuss neoplasia in pathology, and then you'll be able to integrate all this. Where because of tolerance, the malignant cells are self cells, okay? But they're abnormal. We should be able to get rid of them. But because they express self-antigens, then there's some aspect of tolerance where the immune system is not able to properly get rid of them, okay? I won't go to details of, the tumor antigens and tumor-associated antigens. Probably we'll discuss this further next year and see how they've been manipulated for therapeutic intervention. So in a nutshell, that is the scope of immunology and immunopathology. And I will take questions. So what was that question? That comment? I think it was a comment in the chat alluding to autoimmune disease. Any questions or comments? Trying to see if there's anything on the chat. A sperm cell is a foreign cell. Is there any response to it when in a female? And okay, usually there's no response to the sperm cell, but to the majorly not the embryo, but the fetus, because now it expresses paternal antigens. And usually what happens, there's an aspect of fetal-maternal tolerance. There are changes that take place in pregnancy, and you'll cover that in, I don't know when you do OBS in the new curriculum, maybe 4th year. There's some sort of mild immune suppression that takes place, as well as tolerance towards, especially at the placental maternal interface, so that the pregnant mother doesn't induce an immune response against the fetus. How long are immune inhibitory drugs given to the patient? In the context of transplantation, usually as long as, depending on, and we'll talk about this later in transplantation immunology. It depends on the degree of match and mismatch in the HLA antigens between the donor and the recipient. I don't want to confuse you right now. We'll talk about this later. And, if these patients are getting sensitized, either because of infections or because of transfusions and things like that. I will send I think the schedule has already been uploaded. I don't know where, but I think it's supposed to have been uploaded by the administrator, but I'll send a copy to your class email. Yeah. And yeah, the recommended textbooks are indicated on the schedule. So you'll see that there. In some cases, you find that people from the rurals tend to be less sick than those from the urbans. What could be the cause? So what do you mean by sick with what? Is it with infectious disease? Is it with cancer? Is it with allergies? Joshua? Because sick is not just one thing. There's so many things that cause illness. For example, with allergies, it's seen to be more prominent among people in urban regions, and we'll discuss why later. Yeah, I don't know what you mean by which sickness are you talking about? The cats are going to be combined with the rest of pathology. So your cats, there'll be 2 cats, no 3, no 2. I think second year there are 2. An essay cat or short answer question cat and an MCQ cat. So it will include the whole of pathology. You'll have anatomic pathology or general pathology, for you guys, we call it general. You'll have clinical chemistry, and you'll have hematology and blood transfusion. So there'll be combined cuts. I think the cuts will be on, when you get your hematology schedule, they'll be on the date of the hematology. Yeah, so that's the date that we'll use for the cuts. Yeah. Yeah, so I think everything else will elucidate itself as we go further into the actual, the different topics. So everything I mentioned today, we will discuss in detail. There'll be a topic alluding to almost every concept. So I think things will make more sense then. All right, have a good day and welcome to second year and pathology, specifically immunology. Okay, bye.